Now that we've talked about enzymes and enzyme kinetics, I want to talk about some things that are really can be important for how enzymes function. And I mentioned these earlier. There are two things that some different enzymes require, and that one of those is our metal ions. So different metals that we take in by our diet are required for our enzymes to work properly. I'll show you a few examples of that. Also, coenzymes are another kind of thing that we require. These are organic molecules that, again, a lot of times we call them vitamins that we take up in our diet that are required. So, difference between cofactors and coenzymes. The cofactors generally are kind of thought of as metal ions. Coenzymes are these inorganic compounds that can act as a cofactor. So anything that is essentially required for an enzyme to work that's not part of the protein bound by intermolecular forces. So you should be able to, in your own words, kind of describe and define these. But really, a cofactor is more general. Coenzymes more specifically talking about a vitamin. Here's some examples of two different cofactors. On the left is retinol. It's a derivative of vitamin A, and these are the things that are found as ligands in the tissues of your rods and cones in your retinal tissue. So the visible light actually causes the conformational change of the retinal, and the protein that's bound to it will then change as well, and that will lead to opening of a pore within and calcium uptake, and then that activates the signal transduction mechanism, that an action potential that races down the axon and then sends a signal to the brain. So the shape of this is really important in these double bonds here. That's another importance of a cis and trans, because it actually is a cis and trans conformational change that actually activates that protein structural change. Here on the right is a heme binding to the iron cofactor, right? This is a metal that we need. That's why we need iron in our diet. And this is an example of something that's helping transport oxygen within our blood, because the oxygen's attracted to that positively charged iron cofactor. A lot of research has been done on vitamins and minerals and what happens when you don't get them. One of the surprising findings early on in studying nutrition was that people tried to take fat out of a diet, and because they thought, well, fat's probably not really necessary for your diet. Well, in fact, it's very important, because there are a lot of different vitamins that are found inside in these fats and solubilized in these fats. So what they did was a study where they basically, they had animal feed that had protein, carbohydrates, and fat, and they tried to, when they found out that fats from milk or eggs help the animal grow, while fats from lard or olive oil stunted them. So from that milk fat, researchers were able to isolate vitamin A, and they led researchers to methods that helped push nutritional science into the modern era, understanding what we eat and why we need to eat those things. A lot of this nutrition started in UW-Madison. Harry Steenbach, there's a building named after this guy who studied coenzymes and vitamins within our diets. And he actually discovered a way to increase vitamin D content in food, which allowed us to eliminate rickets. Quaker Oatmeal, one of the cereal manufacturers, tried to purchase the exclusive rights to use that method to increase vitamin D for $1 million in 1923. That's $10 million in today's dollars. But instead, they returned the money to university to make future research and discovery possible. So he helped create, with some of the money, that he didn't allow exclusive rights for it, but they used this to actually create this Wisconsin Alumni Research Foundation, one of the largest university technology transfer offices in the world. And so it helped protect that money that came from this discovery, helped UW scientists create more patents and more licensing opportunities. So another way to look at the coenzymes, the vitamins, cofactors, this is more of a broad term that describes non-protein components of an enzyme. We have essential ions, right? So metal ions or activator ions or coenzymes. Prosthetic groups tightly bound, maybe by covalent bonds or co-substrates that are more loosely bound. Co-substrates, an example of this are like the FADH and NAD+. Those are things that are going to be involved in the electron transport chain. For example, they can't be tightly bound because they need to move and interact with other things as well. Where prosthetic groups like hemes, for example, are going to be bound and more permanently attached to that enzyme or protein. A lot of animals actually have lost the ability to synthesize a lot of our coenzymes, and when we lose that ability, those are actually then referred to as vitamins because we have to include those they're vital elements in our diet. So you should be able to kind of look at this and decide which one you think is correct. If you're not sure, go back. Here's an example of the cofactor zinc, and it's important in carbonic anhydrase. You can see here that the side chains, so we have 3 histidine side chains that are bound to this positively charged zinc ion. And so this is important for, it helps to promote the ionization of water. So you can see that the hydrogen came up to this base that's attached somewhere else on that protein from water is here, H2O, but the hydrogen's here. And the positive charge on zinc is able to interact with the oxygen in water so that we can kind of ionize this water. It helps to promote this. And so then this ion then allows the interaction with carbon dioxide. You can see CO2 coming in here, CO2. We can kind of see this is the carbon, 2 oxygens. And then this is positively charged carbon, right, because it's attached to oxygen. So we have this delta positive carbon that is going to then interact with the negatively charged oxygen over here, as we see up here. And then we have this interaction, and then we're able to release this compound from the zinc, and that is now the bicarbonate ion. So we'll pull this off, and then we're left with a positively charged hydrogen. And then we can start the whole process over again, recycle the enzyme. So this relates, remember we were talking about how this happens in our bodies. This is an important enzymatic reaction. We talked about the pH, that case study. So this is kind of, zinc is really actually very important in that process as well. So if we don't have zinc, we cannot take carbon dioxide and turn it into bicarbonate. I mentioned that that experiment where they took out fat and they looked at fat from different sources. And then we were able to identify vitamin A within fat that was important for that component to be, that was missing in the fat. There are actually a number of different fat-soluble vitamins, A, D, E, and K. I like to say A to K. That's an easy way to kind of remember those things. So these are all the fat-soluble vitamins, things that are in fat. Different water-soluble vitamins, the C vitamins, B vitamins are important as well. So there are a lot of different things. These vitamins, the originally came from the vital amines. So a lot of these are nitrogen-based compounds. So it used to be spelled vital amine. They changed it to, they took the E off. So if you ever do spell it the wrong way like this, like I like to do, then you at least can say, well, they used to spell it that way, but they shortened it just to be confusing, like we like to do with a lot of English words. All these vitamins have a lot of common names. So ascorbate is also vitamin C, thiamine, vitamin B, riboflavin, B2, nicotinic acid, B3. So these B vitamins, cobalamin, B12. So, and there are all these different diseases that they're important for. If you have deficiency, you will get these diseases. So scurvy is examples, Beriberi disease. So of all of these different problems that can be caused when we don't have sufficient amounts of these things. Something that B4, you'll notice some of these B vitamins like, oh wait, where's B4? So B4 is actually an adenine base of DNA. If we couldn't make a B4 vitamin on our own, we'd be really in trouble. So this isn't a vitamin, it's not a vital amine. We can actually make that ourselves, so it's not a vitamin. B8 is actually also inositol, It's produced from glucose in the human body. We use that for different signaling mechanisms. So they're not really vitamins. Again, since our body can produce those, we don't have to take them up through our dietary habits. So here's our list of important coenzymes. So some major coenzymes for different metabolic processes. ATP, you'll notice a lot anytime we're involving anything with energy and energy needs. Acetyl-methionine, that's another compound. Some of these vitamins here that are listed on the right. Also, and then you can look through these. These are the various metabolic roles, and then co-substrate, prosthetic group. So it kind of shows you how they're involved. So some of these are vitamins, some of them are not, because if we could make them, obviously they're not. Here's an important co-substrate, NAD and NADP. On the left is the oxidized form, and the difference here is if you have the NAD versus the NADP, this is phosphorylated. So that's the difference between that. And then if we have the NADH or the NADPH, again, it's just this, if you have the P or not, as if it's phosphorylated down here on this second carbon on the bottom of this ring here. This is actually where the oxidation or the reduction happens. We add the hydrogen atom on the top here in the oxidized form or the reduced form. So that's where we add the H if it's the reduced form. The H is removed if it's the oxidized form. These substrates, co-substrates, are oftentimes involved in the oxidoreductases, and they're involved, right? So here, this is the oxidized form, the reduced form. Here is if this is the oxidized, right? So this is reduced, and this is going to be oxidized. We can also see that with that carbon is actually gaining oxygen bonds. That lactate to pyruvate becomes oxidized. So this is an example of a type of metabolic reaction that uses this substrate. And again, it's involved in those oxidoreductase reactions. We can recycle this. It can go through the electron transport chain, and we can use that to create some ATP, which we'll talk about in future. FMN or FAD is another important cofactor, as it's used as a prosthetic group. So again, used in oxidoreductase reactions. These things are derived from riboflavin or B2 vitamin, and they're reduced by taking up a proton and 2 electrons. That's what we're kind of showing you here, the proton and the 2 electrons. And these things are found in the electron transport chain. They can donate a single or double electrons. So unlike the NADH, which only does 2 at a time, this can do 1 or 2. It depends on what kind of chemical reaction that's happening and the different types of oxidoreductase reactions that are occurring at the time. Coenzyme A is an important component of, we can see that the ADP component, this is adenosine diphosphate. So like the base pair, adenosine and 2 phosphates. It's kind of like the backbone of DNA, but it's actually also can be used as a coenzyme in different enzymatic processes. Panthoate form, this is actually generated from vitamin B2. Beta alanine is an amino acid, right? Thiol group at the end to mercaptoethylamine. And so this is actually involved, it's important for the acyl group transfer. A lot of times many fatty acid components have this kind of acyl group involved in the transfer, and we'll talk about that when we get to fatty acid synthesis. Acyl carrier protein, or ACP, it's similar, but kind of similar idea, is also involved in acyl transfer group, but you'll notice that this has kind of a protein backbone instead of this DNA backbone kind of component. Similar prosthetic group here, and again involved in the similar type of process. A lot of times we're not going to show you, or I'm not going to show you this giant, big, huge thing, or this coenzyme. I'm not going to show you this whole thing, or in your book as well. It's going to be simplified as just CoA, and then this thiol group, and then we'll have these acyl groups that will be attached to it. So acetyl-CoA, it will be shown this way. And so whenever we're adding or removing an acyl group, you might see the CoA, or you'll see the acyl carrier protein attached to that acetyl group. Those are different ways that we'll see this. So here's this is ascorbic acid. It can act as a reducing agent in many reactions. So this is where we kind of have humans, bats, pigs. Some primates cannot make ascorbic acid. Some primates actually, some other mammals can. It's a glucose derivative that that many other animals actually can synthesize this. And so this is where the kind of limeys came from, where the sailors found that if they soaked limes in their barrels of water, they were actually able to prevent scurvy from happening on long sailing trips. That's actually an interesting case study here that kind of talked about a child actually would develop scurvy because they were on an almond milk based diet where vitamin C was not incorporated into their diet. It's not very common otherwise. In the United States, we get plenty of not many of these vitamin-based deficiency diseases are actually common because we're able to get a lot of these vitamins easily in our diet in the United States. And we also don't really need that much of those different vitamins because we're able to recycle those components within our bodies once we have them. There's not much that we need to take up every day, for example. This is the process that some animals that are able to create this are actually able to make this L-ascorbic acid, that vitamin C, that we're not able to make. We're missing the GULO gene, GULO enzyme is missing in humans, so we actually can't make, this is the gene that we're missing, going from L-glucolactone to keto L-glucolactone. Here is the structure of biotin, another important prosthetic group. So consider a prosthetic group because it has actually a covalent bond attached to the biotin portion that's generally attached to a lysine amino acid side chain. So we have biotin, and it's generally involved in carboxyl group transfer reactions, usually also involving ATP dependent reactions. You can see this kind of biotin. We have these groups that have a thiol and an amide functional group as well on the side here. In this example, here's a pyruvate carboxylase, turning the enol pyruvate into oxaloacetate. So this is actually the buildup we can create from smaller compounds like pyruvate. We can create bigger compounds, or we add on a CO2 group actually. So we do this through bicarbonate, which we've seen a couple times already, and with the biotin prosthetic group in the pyruvate carboxylase, we can use this to actually bind onto that amide functional group. We bind that CO2 group, and then we're able to use that to add on to that double bond here. So this would be a lyase type of reaction. We're adding to that double bond a CO2 group. Somewhere else that you have seen or that you may have used, or where people use biotin, is actually Western blotting. Takes example of using biotin, this is the big long thing, and avidin to actually undergo that chemical reaction. Once you have your secondary antibody on there, use biotin and avidin to do that color reaction oftentimes. Here's another important prosthetic group. Notice cobalt here in the middle of this heme prosthetic group. We have many amide functional groups on the outside of this, and that cobalt, that ion in the middle. We also have a nucleic acid. Here's the sugar and the base and the phosphate group that's attached to that as well. This is vitamin B12. Deficiencies lead to a lack of red blood cells, not because they're necessary for the heme synthesis, but they have other important functions in red blood cell development, and important for fatty acid metabolism. Here we can see various different hemes, cytochromes, for example, cytochrome B, cytochrome A, cytochrome C. We can synthesize hemes. So it's a coenzyme, not necessarily a vitamin. We can make these in our bodies. oftentimes important for binding different compounds such as oxygen here. You can actually see oxidized versus reduced forms. We can look at this. We can look at these using light spectroscopy for determining what the shapes are, how they're bound to their substrate, oxygen, for this example. So we're going to start talking about some of the fat-soluble vitamins, just looking at specific examples and what they look like. You know, they have a lot of longer hydrocarbons, hydrophobic, aliphatic side chains. They're absorbed similar to the way that we absorb lipids and cholesterol. And remember, we talked about the intrinsic factor, which is released in the stomach, that helps to absorb some of these fat vitamins as well. First one we're going to talk about is vitamin A, or retinol. This has a biological function in the retina, and it's bound to the protein rhodopsin, which actually double bonds are able to receive the specific wavelengths of light, and when they are received, they can change the structure from a cis to a trans, or a trans to a cis, and that change then opens up that rhodopsin channel, releases calcium. It's a calcium-sodium channel, and activates the action potential from firing. So retinoic acid is a paracrine signaling molecule, important for developmental biology processes. So a few different requirements or important functions of these vitamins. Vitamin D, there are different types of vitamin D. It's a family. D3 is formed when sufficient sunlight actually hits our bodies. If you have a deficiency in vitamin D, you can actually get rickets. This vitamin helps to control calcium utilization in our bones. So obviously, if you have problems, or if you have low amounts of vitamin D, you have problems with collagen and bone formation, tissue formation in that area. Vitamin D3 can actually be produced photochemically. The precursor sterol 7-dehydroxycholesterol is present in the skin or the epidermis of man and most higher animals, and sunlight actually allows for the conversion of the previtamin D3, which can then go through further processing to other components, but also the vitamin D3. Although it's important that this is a vitamin, it actually can be endogenously produced or produced in our own bodies, as long as humans or the animal has access to regular sunlight. So that's an important reason that we get out in the sun to some extent and get some sunlight for our vitamin D3 that we need. And then if we get that D3, then there's not ultimately a requirement or a need for us to get dietary supplementation for that. So we have A, D, and then E. Vitamin E is another fat-soluble vitamin. You can see why this would be fat-soluble, right? A lot of hydrocarbons, very long chains that are nonpolar. Vitamin E and vitamin K both look similar with these long kind of fat tails. Vitamin E can function as an antioxidant, can absorb some of those oxidative stressors, and vitamin K is involved in blood coagulation. If we actually overeat vitamin K, you can find vitamin K in high amounts in broccoli. You can actually die from overconsumption of vitamin K. It's kind of interesting. Some of these vitamins, right, you can have too much of them. Warfarin is a vitamin K inhibitor. This is the structure of warfarin. There's an interesting story kind of going back to the University of Wisconsin-Madison also. Warfarin, I talked earlier about the WARF, the Wisconsin Alumni Research Foundation, kind of started by Harry Steinbeck and the discovery of vitamin D. So the Wisconsin Alumni Research Foundation, this drug was developed in part because of that work. It was named warfarin. It's a vitamin K inhibitor. It actually was first used as a form of rat poison. It's a blood thinner. And so people actually take it now for lowering blood pressure and thinning the blood out. But also what it does is with mice, if they eat it, they get small cuts on their feet or their little paws, and they run around and they get little cuts, and then they essentially bleed out and dry out and die. It's also can be used as a blood thinner that people take pretty regularly. So speaking of things, common drugs that people take, you know, the warfarin for high blood pressure. And here's another example of Lipitor, actually, is something that people take for blocking cholesterol. If you have high cholesterol, people can take Lipitor. It blocks cholesterol synthesis, but it can also block coenzyme Q, CoQ10 synthesis. And CoQ10 is important it's an important membrane associated in electron transport chain, so it's involved in the mitochondria and ATP production. Ubiquinone is Q here shown, and then you can have the addition or subtraction of electrons, semiquinone, and then ubiquinol. So these are all involved in carrying electrons on the electron transport chain. Lipitor again can block this. You can kind of see acetyl-CoA, HMG-CoA. We talked about the CoA cofactors as well. And then eventually we can create, from these, we can make cholesterol, but also one of these things that we can make from the acetyl-CoA and cholesterol, kind of the chain earlier, we can actually take off of this and make this CoQ10, which I just talked was important for the electron transport chain. Lipitor is a statin drug. It inhibits this reaction between HMG-CoA and mevalonate, and it actually this is where the statin drugs block. So we're not making cholesterol, which is good if you have high cholesterol, but it's also not good if you need coenzyme Q10. So a lot of people actually say that Lipitor kind of gives them sore bones and achy joints, and you can think about your muscles, how much energy they need, the metabolic requirements of a muscle. If the ATP production isn't happening sufficiently from your mitochondria, because you're blocking this synthesis of coenzyme Q10, you might have those problems. So CoQ10 isn't necessarily a vitamin. We can make this ourselves, but sometimes people get low on those. And it's recommended, if you're taking Lipitor, to also consider getting supplements for CoQ10 to help when you're on those statin drugs, to get enough of this for your muscle production. So that's all the enzymes and cofactors and coenzymes here, so hopefully you learned something from that.